These are notes on how the DNA of your cells becomes the traits that you see. Later we'll talk a little bit about the role environment plays in the traits you see and how it interacts with your genes, but for right now we're just going to stick within the cell. So a brief outline would be DNA to RNA to protein to traits. Obviously there are steps in between. You can take this to different levels of depth. But this is the good basic overview. And if you're following in your book, it's pages 512 and then 521 to 530. So first I want to just review what a chromosome is. We did this a little when we looked at DNA last unit. But remember that a chromosome is essentially a long piece of DNA. So that model we made in class, that would be one chromosome. And I'm going to go this time from the atomic molecular here, so this is the DNA, all the way to the microscopic, so these are the chromosomes. And anytime you see like X's like this, that's actually two chromosomes attached together, and that is what happens before the cell undergoes mitosis. And you can even see way down here at the bottom, it says metaphase chromosomes. So down here it's indicating that. I'm going to get a highlighter or a pen so that you can see that. Yeah, so right here it says metaphase chromosomes. So we understand these chromosomes are undergoing mitosis, which is why they look like X's. It's again actually two chromosomes. But starting on the atomic molecular scale, this is the DNA. And so these colors are representing the different bases. You'll see that red always pairs with yellow, maybe that's the A and T, and then blue always pairs with green, maybe that's the G and C. And so the way this is organized or wound is the DNA is wound around these proteins that are called histones. Okay, now we can get into like, again, there's a lot of detail on this that I'm not going to go into, but the DNA is essentially wound around these histones, okay? And then each set of 8 histones with DNA wound around it is called a nucleosome. The nucleosomes then are kind of coiled together to produce an even tighter coil. So imagine, you're coiling up a string and then you coil the coils. And that's what chromatin is. So this whole thing is chromatin. You can see that's labeled there. We know what chromatin is. And then as the chromatin loops together, that's what a nice bunched up chromosome is. So a chromosome is really just this really tightly wound, very long, neatly organized coil of DNA. But as we're going to see, the reason that only happens during mitosis is because really we need to read pieces of this DNA, right? It's hard to read, like the blue is the DNA. It would be hard to read something in here when it's all nicely coiled. So we want it kind of uncoiled so that we could read, say, like this length there, something like that. So the way the information flows is the DNA is what dictates what proteins are made, and the proteins dictate the traits. Now we've already seen a lot of how proteins help dictate traits, right? We looked in the lab at the lactase enzyme. Enzymes are proteins, and that if your body is making the lactase enzyme, you would have the trait of being lactose tolerant. You can drink milk. If you don't have the lactase enzyme, if your body's not making that protein, then you would be lactose intolerant. We also, we've looked at other enzymes as well, like in the bioflowers, right? Those were kind of fake, but we looked at if you have this enzyme, then you can make this color bioflower. If that enzyme is non-functional, so if your body's making a different kind of enzyme, then you don't get that color and you remain colorless. Also remember when we looked at cell differentiation, we talked not just about enzymes but proteins. So like you should all be familiar with the facilitator protein, which it's a protein that controls what goes in and out of cells. Well, without that, you can't do something like cellular respiration. So that obviously leads to certain traits. It's the trait of a cell that allows certain things in and certain things out. So we understand this protein to traits link. That's already there. What we're really looking at today is the DNA to protein link. Alright, so looking in a little more detail at the chromosome structure, I want to look at genes. So there are regions of chromosomes that are called genes. So this is like one long chromosome. This is chromosome number 7. And it's not, if you were to look at it under a microscope, kind of going back to here, right, you don't see different colored bands on this. This is just our modeling of that, of those genes. So genes are regions of a chromosome which determine which proteins can be made by the cell. So going back to here, right, we totally get how proteins make the traits. Now we're going to look at how DNA makes the proteins and fill in that. So we're almost working backwards from the real macroscopic, what traits do you see, to the atomic molecular, what's going on in the molecule that's then somehow giving us these proteins which give us the traits. This one chromosome contains about 1,800 genes and over 150 million nucleotides. That's in one chromosome. One thing to notice is like this gene here, notice, is encompassed by some blue strands and some white portions. So genes actually, there's coding and then there's non-coding portions on your chromosome. So the coding portions, the parts that code for something, are called exons. You don't need to remember that term. And then the non-coding parts are called the introns. They're in between the coding parts. And I like to remember exons because it's expressing a gene. But what's kind of interesting, and you're going to see this a little later, I think when we talk about mutation, can't remember exactly, but you're going to see that one of the things that has to happen is that all of the exons for a particular gene need to be spliced together. They're not actually all in one long part of the DNA. So like this may be part of the gene here, and then this is another part of the gene there, and those two pieces need to get spliced together. Those non-coding portions, we're going to see some purpose to those later. Just because they don't code for a gene doesn't mean they're useless. So what a gene is it's the order or sequence of nucleotides, which then is a code or instructions that cells use to make specific proteins. Genes don't make proteins. Genes give the instructions or the code to make proteins. So if you were to look, let's say this is a gene, although it would be way too short for an actual gene, but right here, this code here, ATA, ACA, etc. and so forth, is the code or the instructions or the recipe to make specific proteins. So let's now look at this in let's give an example. And if you're breaking these notes into 3 days, now is actually a good time to stop. If you're not breaking this into 3 days, let's continue. So we have our round buds again. We remember these. We have red flowers and white flowers. And just to recap, remember we have this colorless molecule, goes into enzyme Q, turns into a red pigment molecule, right? That's when we have a functional enzyme Q. So now let's bring this into the cells of the flower, okay? So here we have two flowers. We have one that has cells with a functional enzyme Q. So remember, functional kind of means it's doing what it's meant to do. And then we have cells with a gene for a non-functional enzyme Q. And so there's still a gene there. That's something that's really important. There's still something being made, but it's not something that's going to be able to turn the white pigment molecules into red pigment molecules. So the way we model that in a cell is, here's our nucleus of the cell. Remember, the DNA is in the nucleus, and we just draw kind of squiggles. You'll notice they're not X's, they're just squiggles. And then we're going to make a little box here, and we're going to say that's the gene. And just by convention, we color in the functional gene, we leave the non-functional gene not colored in. So that's where, if you were drawing a diagram, that would be the difference. Okay. One thing that is simplified in this model, and we're going to talk about this more in our next unit, is that most flowers actually have 2 copies of each gene. And so they would have a cell could have a functional, like 2 functional copies, or 2 non-functional copies, or 1 functional and 1 non-functional. We're not going to worry about that now, but I just want you to know that this is a simplified version. So in then the cells of the red flower, right, all of this white pigment molecule gets turned into red pigment molecule. And again, I specifically had 6 pigment molecules with 3 enzymes, because remember, enzymes can be reused. Whereas the cells in the white flower, they don't have any enzyme. Now, one thing I don't like about this, and if I were to redo these notes, which I'm probably not going to do for many years because I just redid them, I would have had some sort of non-functional enzyme in here, right? Because the idea is there's still something being made. It just isn't something with the correct active site. Just like the lactase in the lab, there was still some protein in there. It just wasn't something that lactose could fit into to get turned into glucose. Alright, so again, let's go back to this bioflower with the blue flowers and the red flowers. And remember, we had this pathway, so we had a colorless molecule becoming enzyme A, or going into enzyme A and becoming a blue pigment molecule, and then going into enzyme B and becoming a red pigment molecule. And now what we can understand from this is that enzyme A and enzyme B are made from 2 different genes. So there's a gene that makes enzyme A, there's a gene that makes enzyme B, and those are going to be separate genes. They may be on the same chromosome, they may actually not be on the same chromosome. So let's look at all of the options, okay? And again, normally you would have 2 of each of these chromosomes and 2 of these, but we're going to stay simple now. So one option is that you could have 2 functional genes. You could have enzyme A as functional and enzyme B as functional. And so I would actually encourage you to pause the video and predict what's going to happen in that flower, and maybe even model it, throw in some enzymes. So it would look like this, right? Here's our enzyme A, here's our enzyme B, and we get, not surprisingly, our blue pigment, our red pigment, and we get our red pigment flower. Okay? Whoops, sorry. So then we get our red pigmented flower. Alright, so that's one option where both of these genes are producing functional enzymes. Another option is one gene is producing functional enzymes, so let's say B, whereas one gene is producing non-functional enzyme, so for example A. And so here, notice we get our enzyme B, and that's totally functional. It looks, if we go back here, exactly like this enzyme. However, in this case, and I'm going to draw it in this time, let's say we have these non-functional enzyme A's here. There we go. So we're producing something, but it doesn't have the right active site. Now those cannot react to become the blue pigment, which means they can't react to become the red pigment. So the correct enzyme is there. It's being made by this gene that's creating functional enzyme, but because this gene is creating non-functional enzyme, you end up with a colorless flower. There's actually another way to get a colorless flower, and that's obviously if you have 2 non-functional genes, but that would be really, really rare to get 2 non-functional genes that both happen to be making enzymes for the same trait. Okay, so then we have one final choice, and that would be if gene A is producing functional enzyme and gene B is not. So we'd have something like this, and then I'm going to draw in Here's my non-functional enzyme, okay? Because again, we're producing something, it's just not the something that the pigment can fit into, the substrate can fit into. So here we change to blue, but then there's nothing we can do. Of course, with my dumb luck, I ended up putting the enzymes exactly where some of the pigment ended up, but you get the idea. So this is how we would get a blue flower. And so the pathway is exactly what we've already learned, but now what we're doing is we're saying there's actually genes on these chromosomes that are causing the cell to make this protein, this enzyme. And that's then how we get the color. So now, if you're breaking this into 2 days, now is a good day to stop. If you're breaking it into 3 days, this would be the end of your second day. So now we sort of understand in a very general term that, okay, DNA somehow gets us to protein. We totally get how protein gets us to traits. So now we're going to really, really zoom in on how DNA gets us to protein. We're going to look at that very closely so that you can really understand that. So first we're going to look at a cell's nucleus. Whoops, this wasn't meant to be here. Oh well. So much for my brilliant animation. Alright, so looking into a cell's nucleus, here we have a cell. This is the cell membrane out here. Here we have the nucleus. All of the other organelles are in here. I can draw you a pretty little mitochondria if you want. Here's a vacuole. Alright, get a Golgi body. Whoops. Here's our Golgi complex. Okay, ignore that. And then the chromosomes are in the nucleus. The chromosomes never leave the nucleus. You never want your DNA to leave the nucleus. Remember that your DNA is essentially all the instructions you need to make every protein in your body. So you don't want to screw that up by having it go out wandering into the cruel, cruel world of the cytoplasm of the cell. It would be like if you had a favorite recipe and you asked somebody for it. They probably wouldn't give you the original recipe. They'd take a picture and send it to you. They'd photocopy it, but somehow they would give you a copy of the recipe. Well, essentially, what the cell is going to do is make a copy of the gene and then send it out. Oh, actually, the most important one, let's put in some ribosomes here, and then send that copy out to the ribosomes to do the actual building of the protein. So if we're zooming in, we're going to zoom in on one chromosome here, and then we're going to zoom in on a piece of that chromosome, and there's the rest of my DNA, and we end up with this length of DNA. And I've got a key here, so A's are these blue pieces, T's are the yellow, C's are the red, and G's are the green. So this is what we're going to be looking at on the next few slides, is this gene here. Now, no gene would ever be 6 base pairs long. It's going to be thousands to tens of thousands of base pairs long. And to even more than that. I think like the longest protein in our body is 30,000 amino acids, somewhere in there, so that would be 90,000 base pairs. So we're simplifying. Okay, so first what we want to do is we want to do what's called transcription. So transcribing something is an everyday term, but that means to like rewrite it. And so essentially this is the making the copy of the DNA so that you don't lose your DNA, right? The same way you'd make a copy of a recipe. So the way, and this all happens, by the way, in the nucleus. So I don't have the nucleus here. We've zoomed in. Remember this picture here? We're zoomed in on a piece of, so we're like zoomed in on this, that little piece there. So the first thing that has to happen is that the DNA needs to unzip at the gene region. Okay, so just like before we're in replication where we unzip the entire strand of DNA, here we're just going to unzip a little piece because we need to get at it to read it. So we're going to unzip that. And then the next thing that has to happen is there's a kind of RNA. RNA is like DNA, but it's single-stranded. And it also, instead of using thymine, it uses a different base called uracil. So we're going to make our uracil purple. And you'll notice that the uracil and the thymine, they have the same structure. Now, I don't want you thinking that these are the actual structures of these bases, right? These are just models we're using, but I specifically chose something with the same kind of rounded end here, so it would fit into the A here, but then it's slightly different. If you're interested in why uracil is substituted for thymine, I'm happy to go into that either one-on-one or in class, but for those of you who just want to remember that, RNA uses uracil, DNA uses thymine, and then the other big difference is that mRNA is always a one strand, whereas DNA is double-stranded. Otherwise, though, uracil pairs with adenine, cytosine pairs with guanine, it's the same. So we're going to form this complementary strand here. And so you can see it's exactly the same as it is as this strand is here, with the exception of the uracil for the thymine. So now we've made our copy of our DNA strand. Oh, so now, sorry, we're going to jump to the next slide. So now what happens is, oops, okay, and that, I just made this tonight, sorry about that. So the third thing that happens is that the mRNA is going to leave the nucleus and the DNA zips back up. Okay, so there goes the mRNA, it's leaving the nucleus. These black things are ribosomes, so it finds a ribosome. That DNA zips back up, it's happy, it's safe, it's staying secure in its nucleus. So now we're going to zoom in. This is our mRNA strand that we had before. And amino acids are matched to groups of 3 mRNA bases. So we're going to have an amino acid matched here and an amino acid matched here. And those 3 bases together are called codons. That's our name for it. So for example, this amino acid matches to those 3 bases. This amino acid matches to those 3 bases. I specifically made them different shapes because they're different amino acids. And then peptide bonds form between amino acids, and then the mRNA drops out. So there's our peptide bond forming right there. Remember, it's peptide bonds that link amino acids together. This comes all the way back from the beginning of the year when we talked macromolecules. The mRNA drops out, and eventually we will have a long chain of amino acids, which is a protein. So just to show you a less basic picture, here too is the ribosome. So right, we said this happens at the ribosome, but I wasn't creative enough to get a ribosome into my picture. But here's the ribosome moving along a strand of DNA, and so this ribosome would be moving in this direction, going along the strand. This is a special kind of RNA called tRNA for transfer. You don't have to remember that, but that's really how the amino acids get over. So in this picture, the amino acids are different colors, slightly different shapes, and you can see this one is about to link onto that, and maybe then this one will come over next, and that will link onto there, and you get this really long chain of amino acids, some thousands to tens of thousands of amino acids long, and that is of course what the protein is. So we can see how the DNA has provided the template for this, right? This is RNA still. Okay, this is our mRNA. Okay, and this, what I actually don't like about this picture is these bases would just kind of go away. Woo, there they go away. And so, but the DNA provided the template for this mRNA, which is then telling the ribosome which amino acids to add. And going back to protein folding, you know that the order of amino acids in a protein is what determines the structure of the protein, and the structure of the protein is what determines the function of the protein. So now you can tie all this together, where you know that the protein is what's responsible for the trait. You know that the structure and function, or the structure of the protein is responsible for the structure. You know the order of the amino acids is responsible for the structure. You now understand that the order of mRNA is responsible for the order of the amino acids, and the order of the mRNA is based on the order of the DNA. So that's how we get all the way from DNA all the way to traits. So here's where we see that, right? We have the DNA, it's stored in the nucleus, and that gets transcribed, transcription, into mRNA, messenger RNA. That RNA carries a message. So the DNA is responsible for the order of the bases in the mRNA. And then translation, so remember, translating, that's going from one language to another. You're essentially going from the language of mRNA to the language of proteins. And so, right, the mRNA, the order of the mRNA is what's responsible for the order of the amino acids. The order of amino acids is responsible for the structure of the protein. The structure of the protein is responsible for its function, which is what's responsible for your observed trait. So there we see it in this order. On the last slide I said it kind of in the reverse order. This is how you know what the DNA and what the RNA, what the RNA is coding for. And this is called a codon chart. And this looks confusing right now. It's actually not. And some of you are going to understand this way better than you understand anything else, which is not my goal, just to be clear. So let's say you have mRNA and it's U, U, A. So what you do is you look, okay, here's the first base of the codon, that's U. So here's U. So we know we're going to be somewhere in this area. Our second one is also U. Okay, so here's second base of codon. So that's going to be right here. And then the last one is A. I find it easiest just to look down here and say UUA. That's leucine. Obviously, I could go over third base of the codon A and, you know, work my way over here. So that would be the amino acid leucine, also abbreviated LEU. And on some things you'll see that there's just one letter, so like leucine becomes L. What's another one I know? Proline is P. And then I also know that where's phenylalanine? Here's phenylalanine, that's F because proline stole P and phenylalanine starts with an F sound. So in some places you'll see just letters. This codon here, AUG, that's methionine, that's our start codon. Every gene starts with AUG. So that's how the ribosome knows where to start translating, at that start codon. And then we have three stop codons here, UAA, UAG, and UGA. That's how the ribosome knows to stop translating. So even if you get a little extra mRNA than was the exact gene, there's always a start and a stop codon so that the ribosome knows, again, where to start and stop. And then these go back to, if you remember the protein folding, like here's tyrosine. Tyrosine is hydrophilic, and glycine down here is hydrophobic. Those are those same, these are all the amino acids that we saw, and so you remember that like tyrosine being hydrophilic is going to be on the outside of the amino acid, whereas glycine being hydrophobic is going to be on the inside. So remember, that's how that order creates the structure of the protein. And I wanted to show you this. Some of you will like this one better. I actually prefer this kind of codon chart. It goes out from the middle. So before we had, I think it was UUA. And so what you do with this one is you start here with U, and then you go here to U, and then there to A, and again, there's leucine. So I just wanted to show you that because some of you will see that. I prefer this kind of codon chart. Some of you will prefer the other kind. It's up to you. So let's look at the amino acid sequence and kind of work this through, right? So the mRNA is what determines the amino acid sequence, right? And then each codon is a code for one amino acid. So if we have a DNA sequence of this, TAC, CGA, GAT, TCA, then the complementary mRNA sequence is AUG. So same with the A's and T's and the G's and C's with the exception of uracil for thymine. And that's going to be really important let's go back to here, if you're like, I don't know where my T's are, you have to remember, oh yeah, they become U's. So let's come back there. So here's our complementary mRNA sequence. And then here's our amino acid sequence. So the methionine is the AUG, alanine is the GCU, leucine is the CUA, serine is the AGU, and so on and so forth. Alright, so that's a lot of information. Please make sure you are writing down your questions so that we can process it in class and you can make sure you really understand what's going on. Have a good night.